Today, we peel back the layers of a global crisis turning the banana industry inside out. Welcome to the banana apocalypse. This type of banana is the most frequently consumed fruit in the world, and soon it could be gone. I know banana apocalypse sounds both alarming and meme-worthy at the same time, but there is actually a credible threat to banana production. That's why a lot of research is focused on identifying a solution. But first, let's learn more about the problem. The main problem facing banana production is that we grow a single variety around the world that happens to be very susceptible to infection. When a new pathogen emerges that is able to infect this particular variety of banana, it encounters the same genetic individual grown over these vast distances, and that is the perfect opportunity for an emerging pathogen. It can spread like wildfire through cultivation areas. That's why crop monocultures are highly vulnerable to the emergence and rapid spread of disease. Banana is susceptible to many different pathogens. There are many threats to its production. And some of these have caused the eradication of the cultivation of entire varieties. And this isn't the first time something like this has happened. In the 1950s, this variety replaced an earlier one that was wiped out by the emergence and spread of Fusarium wilt. After the emergence of Fusarium wilt in Central America, the spread of this disease had devastating consequences on production. And people were only able to keep ahead of the pandemic by cutting down rainforest and clearing land for yet more banana cultivation in areas that had not yet been affected. But at a certain point, we lost the race. So what can we do? What we're doing is trying to understand where new diseases emerge from. When you see an outbreak in a crop like banana, where did that pathogen first come from? How did it arrive in this agricultural area and start to spread so rapidly? Indonesia is the perfect place to address this question because it's the source of a lot of wild banana genetic diversity, and it's the location of a recent disease emergence event. Plantation-style cultivation is actually relatively new to Indonesia, as it is to many places around the world. And it was soon after the introduction of plantation-style cultivation that people documented a new, strange disease affecting banana. This disease was later identified to be Ralstonia syzygii. After its first identification, the Dutch colonial authorities were very concerned about the possibility of its spread, and they instated a quarantine around the area where it first emerged. And it was in the 1970s that the pathogen, Ralstonia syzygii, escaped this quarantine. And the questions that we're investigating now are, what are the specific origins of this new emerging pathogen? Did it arise via some host shift event from wild banana in Indonesia and then spread rapidly in cultivated banana? And are there, perhaps, some wild banana that exhibit more resistance to this pathogen? We initiated a collaboration with the University of Gadjah Mada and Professor Siti Subandia. She was amazing. So one of the reasons this collaboration is so important and so valuable is that it's a real collaboration. You know, we're equal partners in this work. It's not a matter of someone arriving in a place like Indonesia, taking samples out of the country, and then Indonesian researchers never seeing the benefit of that research. So we traveled and worked with Siti and her doctoral student Adi Prakoso to try to isolate as much Ralstonia syzygii as possible from the field. We arrived in Indonesia actually never having worked with this pathosystem before. And it was a really wonderful opportunity to be able to learn from Siti and Adi about how to spot the disease in the field, how to sample the plants, how to isolate the pathogen. And I'm really looking forward to the results that we're going to generate together and the discoveries that we're going to make together. And this is the best team that I could have ever hoped for. Okay, so how does our science work? After isolating bacteria from banana plants and bringing it back to the lab,
we purify the bacterial isolates by streaking it to a single colony.
We then inoculate a single colony into the media.
And incubate until we have a dense liquid culture.
After that, we extract all of the DNA and prepare for sequencing.
The DNA then be sequenced at the genome center.
So that's the work. But what's the point? I'll show you. After sequencing the bacterial genomes, we can reconstruct the evolutionary history, or family tree, and infer the origin of the outbreak lineage. Once we know where the outbreak emerged from, we characterize pathogen diversity in that area in order to gain a better understanding of what future outbreaks might look like. Got it? Cool. We hope to find the origin of this outbreak. We hope to find resistant banana varieties, and we hope to gain a much fuller understanding of how disease emergence occurs. So banana is important not simply because it's a valuable export crop or because it's a really interesting host to study. It's also important because it's a really critical source of nutrition for many people around the world. So the discoveries we make collectively will contribute in a real way towards improving people's health and well-being around the world.